Well, great. Thank you, everyone, for joining. Again, my name's Alan Cantor from Boston Children's Hospital. And I'm going to begin by telling you about the rationale behind the study and some of the background. And then I'm going to hand it over to Lea Cunningham, who's really going to go through the nuts and bolts of the study. And then we're going to open up for questions for both of us after that. So as many people on the call probably know, we all get 2 copies of most genes, one from your mother, one from your father. And in RUNX1-FPD, it's one of the 2 copies of the RUNX1 gene, which is defective. But importantly, there's one normal copy that remains. And so we've asked the question is, can we find drugs to enhance the activity of the residual, the protein being made from this normal copy of your good copy of your gene? And if we can enhance that, we can hopefully have an impact on the disease, particularly trying to reduce the risk of leukemia. Just to begin with, as many of you may also know, so RUNX1 is what we call a transcription factor. And a transcription factor is a protein that's normally mostly in the nucleus, and it binds to various places in the chromosome, to the DNA, and then turns on and off various genes, and this is how it functions. And on the bottom here is sort of a schematic, a graphic of the RUNX1 protein, and it has different parts of it that do special things. So first, there's this runt domain in red. This is the part of the protein that binds to DNA and recognizes specific places to bind. And there's this blue region called the transcriptional activation domain. This is the part of it that turns on other genes. And then there's this ID, or we call autoinhibitory domain. And this is an important part that actually regulates how active RUNX1 is. And this is what I'm going to be focusing on today. So a number of years ago, we, from work that was supported by the RUNX1 research program later, and as well as some other groups, discovered that there's a chemical modification that occurs in this autoinhibitory domain. It's the addition of a phosphate group to this region, and this occurs by a protein called a kinase. And when this phosphate group is added to this region, it makes RUNX1 less active, or turns it off. But we also knew that there are other proteins called phosphatases that can remove this chemical addition, and this would place RUNX1 in a more active state. We also knew that this is a very dynamic process, so these phosphates are constantly being put on and taken off, and there's normally sort of a balance going on. And this is in some of the work from the RUNX1 research program. We discovered that a drug called imatinib, it also goes by Gleevec, could inhibit this protein, the kinase that's putting the phosphates on. And by doing so, we kind of would shift to the dephosphorylated form, or the form without the phosphate chemical groups, putting more RUNX1 in this active or on state. Another way to think about this is that there's a balance of RUNX1, of some inactive as well as some active RUNX1, but by treating with imatinib, we can tip the balance more to the active form. So what we can really do is try to increase the total amount of RUNX1 in your cells. And as I'll show you in a minute, we can probably get it up to about 1.5 copies. Remember, you start off with 1 good copy. We can probably get to about 1.5 good copies worth. And that, again, may certainly be enough to have an impact on the disorder, particularly our hope is reducing risk of leukemia. And so here's some actual data leading up to this. This is a graph showing on the y-axis a measurement of RUNX1 activity. And we're taking these cells where we do this activity, and we're incubating them with increasing amounts of this drug, imatinib or Gleevec. And if you just focus on the dark blue upper line here, you can see as we increase imatinib, we can increase the amount of RUNX1 activity, getting up to about 1.5-fold, or about a 50% increase. And then on the bottom is shown in green. This is an important control for us, just to show that this imatinib isn't just increasing the activity in our measurement, sort of what we call non-specifically. What it really shows is that all this is happening through RUNX1. So the drug is really working through RUNX1 in this effect. And again, this shows that we can increase the activity around 50% or a 50% boost. So the next thing to do was to really perform what we call preclinical studies. In other words, to take the drug and use it in various models, experimental models, to see if it can really rescue some of the defects associated with having RUNX1-FPD or RUNX1 deficiency. And we've done this a number of ways, and I'm really just going to show you one here. And this is work that we did in collaboration with Paul Liu through the NIH Natural History Study and Anna-Priya Agarwal from Oregon Health Science University. What they did is we obtained RUNX1-FPD patient bone marrow cells from the Natural History Study with Paul Liu, and it's possible some of you online may have contributed to some of these cells. And then Anna-Priya took these cells and can grow them in a petri dish under conditions that produce megakaryocytes. And megakaryocytes are the cells that produce platelets in the bone marrow. And we know that with RUNX1-FPD, there's a defect in making these mature megakaryocytes. They don't mature as well. And so she can grow these either in the absence or the presence of imatinib and then measure how well megakaryocytes are maturing. And the data is shown here on the right. So on the y-axis, we're looking at how mature these megakaryocytes get. And you can see from untreated cells, you get a certain level of this development or maturation. But with increasing concentrations of imatinib, and this is sort of shown a little backwards, so going from right to left, you can see there's sort of a dose-dependent increase in rescuing this megakaryocyte maturation. And this represents the average from 7 different patients with a variety of mutations. And we've had a number of other assays that have all kind of shown the same thing, that imatinib can rescue some of these defects, but I don't have time to go into that today. So we also saw this with a couple of other drugs that are sort of in the same class, but we've chosen to move forward with this drug called imatinib or Gleevec for a number of reasons. So imatinib has been around for quite a while. It was first FDA approved in 2001, so over 20 years. In fact, it made the front cover of Time magazine that year because it was really the first kind of what was called a precision medicine for cancer at the time. It's currently FDA approved for the treatment of 6 diseases, and it has relatively low toxicity, and Lea will talk to you more about that in her part. And we have over a 20-year experience of patients taking this drug long-term. Many patients have been taking it for years at a time. Importantly, we also know that the bloodstream concentrations of imatinib in patients who are taking a standard dose, which is 400 milligrams once a day, correspond to the levels that we've shown improve RUNX1 activity in our experiments. So that indicates that this potentially could work. And then lastly, from a practical standpoint, the patent for Gleevec expired in 2016, and now there are generic forms available. And that's important because that means that this is a relatively less expensive drug, which will be important for anyone taking it long term. So with all this data, we've developed and now opened a phase 1 clinical trial of imatinib in RUNX1-FPD patients to try to enhance the activity of the residual wild type protein, again with the long-term goal of using this to reduce the risk of leukemia, and it should also improve some of the other symptoms associated with this disorder. And this trial opened up at the end of last year, and we're currently enrolling patients. So with that, I'm going to stop sharing, and I'm going to hand it over to Lea to talk about the specifics of the study.
So as you heard from Alan Cantor, all of the rationale for the study so far, so I just wanted to go ahead and go through some general considerations as well as the details of the protocol. And just in general, before joining this or really any protocol, the general considerations for each individual are as follows. Basically, is the research of interest to them as individuals? Are there perceived benefits or risks? What are the logistical considerations? And then in terms of detail for this particular study, we do actually have 2 consents that go along with the imatinib RUNX1 clinical protocol. The first one is what we call screening consent to make sure that people meet basic eligibility criteria, such as platelet counts and that kind of thing I'll go into in subsequent slides. And then once all of those basic eligibility criteria are reached, then there's a separate consent for the actual imatinib administration. And then clinical trials are definitely something for everyone to be thinking about, many questions and making sure that any concerns are addressed. In general, phases of a clinical study typically follow a sequential pathway. Alan Cantor just went through all of the preclinical rationale for the study before medications reach people. And then from there, a further stepwise approach to look at small numbers of people and understand the effects of treatment in humans. And then from there, follow-up phase 2 studies typically evaluate additional safety and efficacy signals. Now, for rare diseases like RUNX1, the 100 to 200 participants in a phase 3 won't happen. And so those later stage studies are designed a little bit differently. But the important things are that, like Alan mentioned, imatinib has been FDA approved in 2001 in 6 additional illnesses, so that if somebody with a germline RUNX1 variant had CML or chronic myeloid leukemia, for example, they would be eligible under those circumstances to receive imatinib. But we're starting under these circumstances back at the phase 1, early phase development to see exactly what effects may be seen in the RUNX1 targeted population. So all that to say that imatinib was approved here for these other indications in 2001, and since then, more than 20 years of phase 4 or post-marketing experience for safety and efficacy have been acquired. So, like Alan Cantor said, it's a very well-characterized medication in general. So in terms of the study objectives for imatinib for folks who have RUNX1 variants, the most important thing is to look at the safety and make sure that what we think we know from all of the other populations is still relevant here, and to also determine the best tolerated dose to make sure that there are not intolerable side effects, because we want this to be something that people can take without major effects on their daily lives. Our secondary objectives include looking for any changes in bleeding symptoms and any clinical laboratory changes. So on the NIH Natural History Protocol, people may be already well-versed in the International Society of Thrombosis and Hemostasis Bleeding Assessment tool, and we'll continue to use that on this study, which is a series of questionnaires about any bleeding symptoms in the past. And clinical laboratory changes, we would want to know if there are increases or decreases in platelet counts, if there are any increases or decreases in platelet electron microscopy, or other studies that are typically drawn. We would also like in the dose expansion, which is the phase 2 part of this particular protocol, to look at blood levels of imatinib. We recently took that off of the phase, the part 1 dose escalation in order to reduce time at NIH. And then there are several research studies that we'll do to look at markers of RUNX1 activity to see what the different doses of imatinib do. So in terms of eligibility criteria, we first do the screening. We're starting the study in age 18 and above. We do want folks to have a platelet count that's equal or greater to 65,000, and for the most part to not have major medical problems. A question that's come up fairly frequently is pregnancy. So we do want folks to avoid pregnancy or to use contraception for 2 months on this first part of the study, and I'll show you a schematic of what that looks like. Now, in terms of the type of RUNX1 mutation, it would need to be either pathogenic or likely pathogenic. So people who had like a variant of unknown significance wouldn't be eligible at this time. And then within that, there's also two different types of pathogenic or likely pathogenic RUNX1 variants. There are those that cause haploinsufficiency or half the activity, like Alan Cantor showed on his diagram, but they also don't have a dominant negative effect in the sense that they're not going after the normal copy of RUNX1. So this is something that the genetic counselors, either Natalie on the natural history protocol or Yi Lu on this protocol, can help us determine. And as I mentioned, the history of bleeding or abnormal laboratory studies would need to be known as a baseline. We also want to make sure that people are coming into the study with a baseline bone marrow within 9 months of enrollment, and meaning that anybody who already has a blood cancer, for example, wouldn't be eligible for this trial. And we also want to make sure that any additional mutations that are found on what we call the TSO 500 panel, which is a panel of 500 additional genes that we look for in the marrow, are not problematic at a level of 5% or greater at this time. And then folks would also need to be able to swallow the imatinib pills. In terms of the study participation timeline, it's welcome, to approach any of the team members for any questions or concerns anytime. We can also share copies of the consent forms that have additional detail and schedule phone calls or telehealth to discuss further on an individual basis. We are noticing that people who are here on the NIH campus anyway for their natural history protocol visits, it's usually easiest to also meet with the RUNX1 imatinib study team at the same time. And then we're also able to add on all of those screening labs that I mentioned at the same time. And those include routine serologies to look for things like hepatitis, and to also get an electrocardiogram, which is putting stickers on the chest to look at the heart rhythm. And then again, that TSO 500 sequencing test. And then if it turns out that folks are interested and eligible after all of the screening is done, then we would need to have them return to NIH. And then it depends on how long they want to be here. So for the phase 1 arm, the dose escalation, which I'll show you a picture of on the next slide, people can fly in and out even on the same day if they're choosing not to donate a marrow, for example. And the rest of the study visits can either be done at home or are optional. And just to clarify, at home, we do provide LabCorp slips so that nobody would have to pay a copay or go to their home doctor for that. So this is the kind of picture that I was talking about before in terms of what is a 3+3 design. And I'll go ahead and walk everyone through that, starting here at the purple star. So we're starting what we call an arm 1 dose escalation, and we're increasing the dose starting at 25% of the approved dose, or 1/4 of the approved dose. So just as a reminder from Alan Cantor's slides, the approved dose of imatinib is 400 milligrams to 600 milligrams daily, but we want to give people the lowest possible dose that we can where we're seeing an effect. And so we're starting at 1/4 of the dose at 100 milligrams. And we would be treating 3 people, for example, to start at 100 milligrams per day. So each of these little blue dots here represents a person. And the blue dot means that there doesn't happen to be a side effect. So walking through this, we would treat 3 people at 100 milligrams. And then we treat them for 28 days. And then we watch them for 28 days. And if they have no side effects, then we enroll the next group of people at 200 milligrams. So 1, 2, 3 here, and then we watch them for 28 days. And I threw in, a little curveball here. So one person here at 300 milligrams has a side effect. So if that happens, then we would enroll 3 more people at this dose level and see if that is possibly related to imatinib or not, and then we watch everybody again for 28 days. So if it turns out that 400 milligrams, is the correct dose, then we call that the maximum dose that we find in arm 1. And if that's the case, there's a second part of the protocol that all of these folks here can join potentially the arm 2 expansion cohort if they want, but also it's also completely fine for them to do their 28 days of taking imatinib and their 28 days of observation and say, okay, I've done my part, I'm good. But again, for people who don't have side effects who wish to continue for any reason, then they may be eligible for the arm 2 expansion cohort. And at this point, we would treat for 12 weeks to see, again, if there's any differences in any of the labs or anything else. So this is the imatinib drug information sheet that is widely available in many different places. And the important thing about the drug information sheet is it's really published for the 400 to 600 milligram dose that is FDA approved to treat things like chronic myeloid leukemia, for example. In our study, we're starting at 100 milligrams, so we don't know which side effects we would see. So I'm just going to make a few points here that all drugs have the potential to cause side effects, and what are the potential side effects of imatinib. So when side effects are submitted to the FDA, they basically write down everything that people report, whether it's related or not. It's important to cast a really wide net, especially in early phase studies, to see what might be out there. And so, for example, some people report constipation, some people report, diarrhea, some people report stomach pain or upset stomach. So what I've written here in black is the potential side effects that are reported, and I've also written, if we have a medication that helps those side effects, like ondansetron or Zofran, then that's something that's listed here. Muscle spasm, we would check hydration and electrolytes. If people are feeling sleepy on it, then that's maybe somebody who would want to take it before bed. If people have trouble sleeping on it, then sometimes people respond to melatonin. There are medications, for gas called simethicone. Headaches can be treated with Tylenol, dry skin with lotion, and that kind of thing. But I think the most important take home of the slide is that we don't know exactly what each person may experience as an individual, which is why it's so important to have frequent communication and to we can either treat side effects, or if they're intolerable, then that person would definitely need to stop taking imatinib. But again, we're starting at 100 milligrams. And so what I'm hoping is that the lower doses won't see much of this. So in terms of protocol enrollment, we've minimized how much time is needed to come to the NIH as much as possible. We recognize that people definitely have their own very busy personal and occupational lives. As I mentioned, most of the study visits are from home or on telehealth, and the labs can be done at LabCorp. No insurance or copays are required. Both clinical care and consultations and monitoring are provided as part of the study. I think people who have been here to NIH before know that there is assistance with travel, lodging, and meals, and we can talk about the details about that for the study as well. And we do hope that the participation in the study will identify the best design for follow-up. So in terms of just kind of practicalities, this is a picture of the NIH campus for anyone who hasn't been here yet. This is the Building 10 Clinical Center here. We do have for young adults and adults, there's the Safra Lodge, which is like an on-campus hotel. And then for also some young adults would be eligible to stay at the Children's Inn. Here are the study contacts. Like Amanda said, Rebecca Alexander is the research nurse on this study. Yilu is the genetic counselor on this study. I'm the principal investigator of the study, and Alan Cantor can be contacted with any questions about science background and rationale. And then most importantly, nothing that we do here would be possible without the patients and families and referring providers in the RUNX1 research program for helping to bring everybody together. And in addition to the outside collaborators that you've heard a little bit about, there are others who are very active, in this area. Many thanks to Paul Liu for the ongoing natural history protocol and everyone who's contributed on the natural history protocol. Everyone at the National Cancer Institute who is active in the trial. Everyone in the Department of Laboratory Medicine who, they look over all of the aggregation and all of the bone marrows for everybody. And these are all the folks in other people in benign hematology, gastroenterology, in dermatology, gynecology, pulmonary, and the pain team, for anyone who needs consultations in those areas. And they've all seen many folks with RUNX1. So with that, I will thank you all for your attention. And I'll stop sharing, and then Alan and I would be happy to take any questions. Thank you so much.
Thank you. Thank you both. Those were great, very clear presentations from my perspective. So good job. Okay, so as we typed in the chat, please feel free to hover over the bottom of your screen and click on the Q&A icon to type in any questions that you have for any of the team here. And again, we'll take patient questions, clinician questions, scientist questions. And I will start off with a few. I jotted a few down that I remembered patients having last time or others audience members having when we offered this a month ago. So first of all, in terms of eligibility, there have been a couple of questions around, how do I know if I think I might have a somatic mutation? How do I know if I can still participate or not? And similar to that, how do I know if I have a dominant negative mutation that will just preclude me from participating?
Sure, thank you so much. I can, start off, and also Yi or anyone else can also, jump in. So probably the dominant negative versus haploinsufficiency question is usually determined in several different ways. For some folks, this has been published in the literature. We already know what type of mutation it is. For others, it's still an active area of investigation. So Paul Liu and others have functional assays that they are doing in their labs to help us understand that information. And they're more than welcome to weigh in if they have anything to add about that. As a phase 1 study, we're very stringent in terms of the that criteria. So if there's any question about whether something is potentially haploinsufficiency or dominant negative, then they basically would stay on the screening protocol until we have definitive data either way. I can say that for folks who have a deletion, then that's usually the easiest person to enroll, because by definition, it's not there, so we know it's not going to be a dominant negative. In terms of the secondary mutations, we do the TSO 500, and that tells us if somebody has a secondary mutation, and if so, what the variant allele frequency is, and that's what we call a CLIA-validated test, and so that information is known to the patients and the providers. But I'll stop there in case you'd like to add anything, or anyone else.
Sure. So if you have a report of your genetic testing result, don't feel hesitated to email us, and we will take a look, and then we will give you from our assessment which category it will belong to. And also, if you know you have a secondary variant, and we can don't hesitate to send to us as well. We will take a look, and yeah, the main thing is that you can reach out to us, and we will do our best to answer your questions. Thank you.
Thank you. Actually, yeah, I think maybe Dr. Cunningham, you said, but who's the best first person to reach out to? So if we hear from patients that they're ready to discuss this, who's the first point of contact? Is it Rebecca?
Sure. So yes, Rebecca is a great person to contact. Absolutely. And Yi and myself are also available.
Okay, perfect. We'll send, after this webinar, we will send out the recording slides, and we'll make sure we include the flyer, which you'll see in the chat as well, that you all created about the study. I believe it has everyone's contact information there, but we'll also make sure we send out email addresses for you all as well. There was also a question about age, in terms, so you had mentioned great, older than 18, which makes perfect sense. But someone had asked about whether, the sooner the better, the younger the better. Any thoughts there?
Yeah, that's a really good question. So, as has been figured out over the years that, folks with germline RUNX1 mutations typically have an earlier onset of their clonal hematopoiesis of indeterminate potential. And it is, conceivable that the older people get, then the more likely they may have a secondary mutation that would make them ineligible on this part of the trial. Honestly, this is, one of the most exciting, times to be researching, and being involved in translational and clinical care for folks with RUNX1 mutations. And the data is, every day there's something new that's coming out. And so from some of the other inherited bone marrow failure syndrome fields, we know that some of these mutations may actually be helpful in terms of helping to resolve blocks in blood cell development. And we're in the very earliest stages of figuring out which ones may be helpful and which ones are harmful. So because this is a phase 1 study, we kind of just put a blanket, stop on enrolling people, with secondary mutations until we know more. But I do think that as time goes on, we may be able to safely enroll, people who have some secondary, mutations depending. So stay tuned for that.
So relatedly, so you asked about or you mentioned the phase that we're in on this trial, or you're in. What are you able to anticipate sort of how long you're going to be in this phase of the trial, and sort of what you're thinking for? Obviously, you need these outcomes first before you can say what's next. But sort of what you envision for the next phase and when that might happen. Is that possible to estimate, or does it just depend on how quickly you have patients enrolling?
You're exactly right. So for, really, it will depend on how quickly we can enroll and finish this trial. And that will give us the information on the timeline, for the follow-up.
Perfect. And what makes it a phase 1B versus, a phase 1A, I know you had the diagram, which is really great, about the various phases, but 1B versus 1A, is there a C?
Thank you for asking that. It's that, we have a little bit of information already in hand about the drug that's been in people before, versus starting at the very, very beginning.
Great. Okay, great. Okay. Another thought that I'd heard from others is, so if they participate at this point, will that disqualify them from participating in other trials or future phases of the series of trials? Or will they be able to continue? Is it hard to say at this point?
Yeah, that's a really good question. So, for example, like let's say somebody enrolls, next week on 100 milligrams. So they would take imatinib for 28 days, and then we would watch them for 28 days, and then they're done. That's it. Most other clinical protocols that are in development usually exclude people who have had investigational medicine within 28 days, for example. But being on this protocol shouldn't disqualify anyone from being on other trials should they open in the future.
Okay.
Including a longer phase 2 trial for this drug too. Yeah.
Excellent. Thanks, Artie. Thanks, both of you. There is a specific question for an individual wondering if having stage 1 uterine cancer would block a person from being able to participate in this study. Or maybe you can speak to cancers in general, or other health issues.
Right. Yeah, so that's a really good question. So as the eligibility criteria is written now, not necessarily, but that would have to be, something kind of an individual discussion with the individual person.
Great, thank you. And I was wondering if you were going to mention sort of a coordinated discussion with their home physician who's caring for them, and that makes me wonder actually whether that's something that you'll be doing generally throughout this trial, how you're coordinating with any hematologists or oncologists that patients work with at home.
Yes, absolutely. I think the success of this or any trial is really dependent on outstanding communication with everybody who's involved, the patients, the families, the referring providers, everyone, in the community. Absolutely.
Dr. Cantor, you described a lot of the preclinical work you've done here. I'm curious about where you're going with this yourself, given that a lot of this is now the clinic and at the NIH, and you're in Boston. Are you what is your role in this ongoing? And then are you in the lab working with imatinib still, or what are you up to in this area?
Yeah, we're still we're really focusing some on the mechanisms, why RUNX1 deficiency leads to the leukemia, trying to understand that, looking at different aspects of it, and how medicines like imatinib and similar drugs could overcome that or help address that.
Yeah, okay.
Yeah.
Do you see this coming to sorry if I interrupted you. Was that, yeah. Do you see this coming to your site as another location, or will you all really focus in on having this mainly at the NIH as the main site?
I think for the phase 1 study, we'll probably just keep it the NIH, but our vision for a longer study, phase 2, is, phase 1 is successful, would probably be multi-site for practical reasons.
And of course, you work with children, right? Mm-hmm. So I don't know if that's in the cards for a future phase. Do you know, or is it too early again to say when you would move in that direction, if at all?
Yeah, I think we would probably reduce the age for our phase 2 trial. And I think we would also look at patients who are known to have larger clones, somatic clones, that we would follow.
And relatedly, in terms of location, there have been a few questions, and I know we've had patients sign up for these webinars who are international, and they've been curious about whether they're eligible and how that works with travel coverage and whatnot.
Thank you so much for the question. So it basically works the same way as it does on the natural history protocol, that NIH can help with travel once people reach the U.S. border, but getting to the border is not something that NIH can pay for.
Thank you. Let's see. And then we had another question that asked about, you had mentioned with some of Lea, Dr. Cunningham, when you had mentioned some of the concerns of side effects, I guess is what they were. You had mentioned headache and Tylenol as an option, and I believe some patients have mentioned being advised not to take Tylenol with RUNX1, or I wonder.
Yeah, I'm really glad that you mentioned that. So very strict contraindications to headache type medicines include things like aspirin, ibuprofen, things that fall into the nonsteroidal anti-inflammatory category. It's very interesting that when people come to the NIH for their platelet aggregation, it is something that the platelet aggregation people have noticed that Tylenol can have an effect on that particular study. But unless there are other contraindications for that particular patient, if they have any liver issues or anything else, then Tylenol is usually something that we actually recommend people take for pain. But again, that would be like an individual question.
Okay, so I think we might have addressed all the questions. Do you all want to add anything before we give people a little time, or I'll watch the Q&A as well, before we give time back to everyone, and we'll be able to send out all of this information so people can think about it and come up with questions and reach out to you. Any other thoughts that came to any of you while you're...
Yeah, I was just wondering, maybe I could just clarify one thing. I know Lea had mentioned that we want the patients to have had a bone marrow within 9 months of starting the trial, but we can time that with, an annual surveillance bone marrow, such as on the natural history study. So it wouldn't involve an additional bone marrow, just your usual one, as long as it's within 9 months of starting.
Right. Yes, and I think a lot of our community have, are traveling to the NIH pretty regularly. So it's really nice to hear that you're able to coordinate that and work so closely with Dr. Liu and team. So that sounds good.
One other thing I should mention too, this is really a safety trial to see it, make sure it's safe and determine the dose. But some of the things we'll also be looking at is looking for evidence that it really is increasing RUNX1 activity from the patient, peripheral blood samples over the course, to provide more evidence that this is doing what we think it's doing.
Great. Yeah, thank you for that clarification, because we've been trying to make sure we at RRP have made sure patients are understanding what a 1B trial is, but that's really an important point. It's not just safety. You're also looking at that on as a secondary objective, correct? Okay, great. That's exciting. Well, we're really excited about this trial. It's I joined RRP 5 years ago, soon after Katrine, and we thought, oh gosh, it's going to be a long, long road right towards the trial. So we're really thrilled that this is launched, and here we are, ready to have patients participating and learning, and hopefully we'll get to some treatment soon. Thanks, everyone. Thanks for your time. Let me see if I've got one more. I'm getting questions in different formats. I think I can let you go, and we will send out again the recording, the slides, and the flyer, so everyone has information about this. And if you need to follow up with other questions, you can do so by email. And thanks, everyone. Have a great Friday and a great weekend.
Great. Thanks again for
Thank you,
joining.
everyone.